Welcome to the third episode of my YouTube course, Introduction to Biotechnology. In this episode, we'll explore some key aspects of plant biotechnology and differentiate between two important concepts. We've been building our knowledge step by step, so following the episode order is essential for better understanding. Today's focus is on genes and their role in modern biotechnology, particularly in plants. Don't think of genetic engineering as a single method. It's a group of techniques that we'll continue exploring, including in the molecular techniques lecture. When we develop genetically modified plants, GMPs, also called genetically engineered plants, we're applying modern biotechnology to serve humanity, either by improving agricultural sustainability or developing crops with specific traits. Looking ahead, with the global population expected to hit 9 billion by 2050, the FAO predicts we'll need 70% more food. Plant biotechnology will be crucial to meeting this demand. Biotechnology can be broadly classified into plant, animal, and microbial biotechnology. In the modern context, especially in plant biotechnology, we often deal with genetic engineering, working directly with genes. For example, some bacteria enhance plant growth. These are known as plant growth promoting bacteria, PGPB, a term worth remembering. We also touched on earlier biotech applications like biofuels, which are still relevant, but today's spotlight is on genetic modifications. Modern plant biotechnology allows us to use bacterial genes to develop plants that resist fungal diseases by modifying their genome to enhance resilience. Here's a practical example. Some bacteria can kill insects. Certain insects damage plants like corn, so scientists isolated the insecticidal gene from these bacteria and introduced it into the corn plant. The result is a corn plant that can kill pests, protecting itself. A real applied case of genetic modification. This brings us to a key distinction: gene editing versus genetic modification. Think of gene editing like editing a photo, adjusting brightness or removing blemishes without changing the photo's core. Similarly, in gene editing, we alter existing genes, perhaps turning one off or modifying its function. Take mushrooms as an example. A specific gene causes browning, which reduces shelf life. By editing the mushroom's genome to inactivate this gene, the enzyme responsible isn't produced. The result, mushrooms that stay fresh longer. In 2016, the first CRISPR-edited mushroom was approved for human consumption. In contrast, genetic modification is like adding a new element to the photo, something completely foreign. It involves introducing a gene from another organism to give a new trait. Revisiting the corn example, inserting the insect-killing gene from bacteria into the corn plant is genetic modification, because we're adding a trait from a different species. Another example, if a person has brown eyes and we use gene editing to turn them green, we're just modifying an existing trait. But if we add a gene for wings, which humans don't naturally have, that's genetic modification, introducing something completely new. Summary: gene editing, altering or disabling existing genes. Genetic modification, adding genes from a different organism to create new traits. Being able to recognize the difference between the two is key. Ask, are we modifying something already there, or adding something new? I hope this clear, audio-friendly explanation helps clarify the concepts. In the next episode, we'll build on this knowledge with more examples and deeper insight.